Welcome students. Today our topic for discussion is the new unit, that is the biotechnology. Now what basically biotechnology is, it is simply using the living organisms in a controlled manner for human welfare. As you have seen over here is written that using living organisms in a controlled manner for human welfare is called as the biotechnology. The term biotechnology was first of all used by Carl Urke. This is a Hungarian scientist. A Hungarian scientist first used this very term, that is the biotechnology. So the term biotechnology was given by Carol Erke. Now you see what we do basically in biotechnology, or what basically biotechnology stands for, is that you can use some organisms like the bacteria. You can also use the fungi. You can also use the algae, or you can also use some other organisms for human welfare and benefit. So this very type of technology which is used for the organisms to be used in human welfare, this very technology is known as the biotechnology. As you have seen that the term biotechnology is basically formed by merging two words, that's biology and technology. So it involves technological process in biology by which we can use the living organisms for our own welfare and benefit. So now there are two core technologies involved in this very process. There are two core technologies. There are two core technologies. There are two core technologies in biotechnology. In biotechnology, there are two core technologies. It means that biotechnology stands on two core technologies. The two core technologies of the biotechnology are as, that is the genetic engineering. That is first the genetic engineering, which you sometimes also call as the recombinant DNA technology, simply or DNA technology, or recombinant DNA technology. So this is the first technology, or the first process involved in the recombinant DNA technology. Then another is the biochemical engineering. Another is biochemical engineering. So these are the two core technologies. These are the two core technologies of the biotechnology. First one is the genetic engineering. The second one is the biochemical engineering. Let's first of all discuss what is genetic engineering or recombinant DNA technology. You see, this science was started by a scientist known as Paul Berg. That's why Paul Berg is sometimes commonly known as the father of father of genetic engineering. Father of genetic engineering, or father of recombinant DNA technology. This very person is known as the father of recombinant DNA technology, sometimes also known as the father of this genetic engineering. So Paul Berg is the father of genetic engineering, or he is the father of the recombinant DNA technology. So what we do in recombinant DNA technology and what we do in biochemical engineering, or what we do in genetic engineering and what we do in biochemical engineering. Now, this very system deals with the gene manipulation process. It deals with the gene manipulation process. Gene manipulation process means, for example, there is a gene in case of humans. For example, I will draw over here a chromosome of the humans. This is a chromosome of humans. In humans, there is a gene known as the insulin gene. This is the insulin gene. Now, what we do, you see, many of the humans suffer from the lack of their insulin hormone in their body due to many reasons. Now, how we supply the insulin to such persons? What we do, we excise or we cleave this very gene. Cleave means we break down this very gene. We take out this very gene from a human chromosome, and then what we do, this is a bacterial cell. This is a bacterial cell. Inside the bacterial cell, you will see this is the main genetic material or nucleide of the bacterial cell, and this is the circular double-stranded closure DNA molecule in case of the bacterial cells. These are extra chromosomal genetic materials, and they are known as the plasmids. Now what we do, we take out this very plasmid inside the laboratory, and then we cut this very plasmid, and what we do, we cut this very gene from the human genome, and then this very gene is ligated with this very piece of the DNA, that is the plasmid, and a new DNA is formed, which you call it the rDNA, means recombinant DNA. Recombinant means it has been formed by recombining the DNA of the two different organisms, that is that of the human and that of the bacteria. Now what we do, this recombinant DNA technology, by this rDNA technology, this rDNA is again supplied inside the bacterial cell. Now when the DNA replicates that of the plasmid, the insulin gene which has been ligated now with this very plasmid, it also replicates, and when it transcribes this very DNA or DNA, when this transcribes, the DNA of the insulin also transcribes, and when the proteins are formed by this plasmid DNA, also the insulin gene also forms a protein which forms a hormone known as insulin. So over here we have used this very bacteria as a factory or as an industry for the production of the insulin by the process of the gene manipulation. And this process of gene manipulation, in which we deliberately manipulate the genes for our benefit, we alter the genes for our benefit, we take gene of one organism, then ligate it with the DNA of another organism, or we do some additions, alterations, manipulations for our own benefit. This very type of the technology, or this very type of the process involved in the biotechnology, is the first process that's known as genetic engineering or recombinant DNA technology. Again, I shall be repeating why it is recombinant DNA technology, because we are using the DNA of two different organisms and we are combining it. That's why it is recombinant DNA technology. Then there's another process, that's biochemical engineering. Since this is a complicated process, it's not very easy. It's very easy to say and it's very easy to portray over here on the board such things, but it's very difficult when it comes to the lab or when it comes to a laboratory procedure involved in this. Inside the laboratory, what happens, you have to take all these actions inside the laboratory, and when you when you this implant this very recombinant DNA again inside the bacterial cell, the bacterial cell needs to be cultured. For example, this is a bacterial cell. Why do we do this very whole process? We do this very whole process inside the lab, and it needs later on to be cultured. And for example, this is a culture dish, and this is a bacterium which is to be grown inside there, or any other organism which is involved in the recombinant DNA technology. It is to be grown inside the laboratory, inside the culture medium, and this process or phenomena in which the living organisms are grown, particularly the microbes are grown for recombinant DNA technology. For the purpose of recombinant DNA technology, these microbes are grown inside the laboratories involved in the recombinant DNA technology procedures. This very technique is known as biochemical engineering, in which we grow these very microbes on artificial growth media. Is it okay? So these are the two core technologies. Now we will be discussing what are the different tools involved in the recombinant DNA technology. So our next step in discussion is the tools involved in the recombinant DNA technology. Let's see which type of tools are involved in the recombinant DNA technology. So let's move on forward. So our next point of discussion is the tools involved in rDNA technology or recombinant DNA technology. So it is the tools involved in rDNA technology, means recombinant DNA technology. So what are the tools involved in the recombinant DNA technology? We shall be discussing about that. First of all, what we say that we need to open up the cell. For example, this is a bacterial cell. This is a bacterial cell. Again, I will draw a bacterial cell over here. This is the man genetic material of the bacterial cell. This is the plasma. We need a gene from a bacterial cell, or we need a gene from other organism to be ligated with the DNA of the bacteria. Now, this is the cell wall of the bacteria. You all know that the bacterial cells have a cell wall, and the cell wall of the bacterial cell is made up of material known as peptidoglycan, or that's also known as murin, which is a complex carbohydrate structure. Now, we need to open up this very cell. First thing is, we need to open up this cell. Open up this cell. The first process, we need to open up this cell. For opening this very cell, we need some enzymes. Clear? Then there's another thing. When we opened up this very cell. Or it's not necessary that there is only bacterial cell which needs to be opened up. For example, this is the human cell. The human cell also needs to be opened up, or any animal cell or plant cell. Though human cells or animal cells do not have any cell wall, but we need to open up the cell. Okay, so this is the genetic material over here. For example, this chromosome. We opened up a cell by the help of enzyme. Which type of enzyme we shall be later on discussing. Now, when we open the enzyme, when we open the cell, what happens later on? This is a chromosome, this is a gene over here. We need to cleave this gene, or we need to cut this gene so that this gene can be extracted from this very chromosome, and this can be implanted in this very plasmid. Okay, now, but how we cleave these genes? For cleaving genes, for extracting genes, for extracting or cleaving genes, we also need for extracting or cleaving genes, we also need enzymes. So this is another category of the enzymes. Now, I mean to say over here that we have cleaved this piece of DNA, this is a gene, and this is over here a plasmid. We have cutted this plasmid. हमने इसको cut किया है. We cut this plasmid, or we cut this very gene by the help of enzyme. Which type of enzyme we shall be later on discussing. Now there's another thing. We need to. This is now foreign gene. This is known as the foreign gene now. Foreign gene. The gene which we inject from over here. This is known as the foreign gene of interest, or foreign gene simply, or it's also known as sometimes passenger DNA. So we need to ligate it over here. We need to bind this very DNA of plasmid with this very DNA. That's the foreign gene. Like this is a plasmid, for example. This vehicle DNA also known as. So over here we have ligated with it another what we call as the gene. So for that very purpose, what happens? We need ligation. Ligation means we need to ligate this very DNA, and for that very process, we also need enzymes. So we need enzymes at each and every step over here. हर एक step पे हमने जो है, we need enzymes. At every step we need enzymes, and for opening up the cell we need enzymes. For extracting the gene we need enzymes. For ligating the genes we need enzymes. Now, what are the different types of enzymes which we need? We shall be discussing different types of enzymes. For example, over here, if we need to open up the cell, for example, if we need to open up the bacterial cell, we need a type of enzyme which we call as lysozyme. Lysozyme. Lysozyme is used to dissolve the bacterial cell wall so that the bacterial cell opens out and we can extract the DNA from it. Similarly, then we need to cut the DNA. For cutting the DNA, we need enzymes which we call as the restriction enzymes. Then there are now different categories of restriction enzymes which we shall be discussing later on in detail. After that, we need over here enzymes for joining the different DNA molecules during recombinant DNA technology. They are known as ligases. Those are DNA enzymes are known as ligases. Now there is another category of enzymes which are known as synthesizing enzymes. For example, this is a DNA molecule. This is a simple DNA molecule. We need to generate one more molecule of DNA from it. We need an enzyme which we call as the DNA polymerase. We need an enzyme which we call as DNA polymerase to form another DNA molecule from it. Okay, this is a template DNA. We need to form another DNA molecule from it. It is known as DNA polymerase. It's a synthesizing enzyme. Sometimes there is RNA and we need to form DNA from the RNA. And we call that very enzyme which is used to get DNA from the RNA, that's known as reverse transcriptase. And this very enzyme is also a what we call as synthesizing enzyme. Now we have understood over here 4 categories of enzymes. The first category of enzymes is the cleaving enzymes, the opening up enzymes like the lysozyme, which is used to open up the bacterial cells to get the DNA. Then there are cleaving enzymes which are used to cleave different DNA molecules and genes at specific positions. They are known as restriction enzymes. We shall be discussing. We have to discuss restriction enzymes in detail in the next upcoming lecture. So this general discussion about these very enzymes. Then we need to bind two different DNA molecules with one another, and the enzyme involved in that is known as ligase. which is used for ligating two different DNA molecules to generate a recombinant DNA molecule. Okay, then there are synthesizing enzymes. When we need to form DNA from the another template molecule of DNA, we call that very enzyme as DNA polymerase. Similarly, when RNA molecule is used as template and DNA is formed from the RNA molecule, the enzyme is known as reverse transcriptase. In the upcoming next video, we shall be discussing in detail about the restriction enzymes. There are three different categories of restriction enzymes and how they work and which of them is involved, where we shall be discussing in our next lecture.